The small intestine is going to finish chemical digestion, and it's responsible for most of the nutrient absorption. So the ingested materials spend at least 12 hours in the small intestine as chemical digestion and absorption are completed. What's pretty amazing is that about 6 meters or 20 feet long in an unembalmed cadaver, but much shorter in a living individual due to the muscle tone. So it's quite long. This diagram is showing the gross anatomy of the small intestine. So notice that the first part of the small intestine is the duodenum, the second is the jejunum, and the third is the ileum. After the ileum, then foodstuffs or what's remaining will travel into the large intestine. And then the large intestine eventually will end up at the anus. The duodenum is going to begin at the pyloric sphincter. So the pyloric sphincter is found between the stomach and the duodenum. The next section of the small intestine is the jejunum. It's the middle portion of the small intestine, and it's the primary region for chemical digestion as well as nutrient absorption. The last segment of the small intestine is called the ileum, and the distal end of it terminates at the ileocecal valve, which controls the entry of material in the large intestine. So the ileum, specifically at the ileocecal valve, is going to be between the ileum and the colon, which is the other name for the large intestine. When we look at the histology of the small intestine, we'll see that there are mucosa and submucosal tunics that are thrown into specific folds called circular folds or plicae circularis. And these, there are microscopic finger-like projections called villi, which can be seen on the surface of the circular folds. These play a very important role because they increase the surface area. So there's a greater area on which the broken down particles from the foodstuffs can be absorbed. Then on top of the villi, there are also finger-like projections called microvilli, which can be seen on the surface of the villi. And collectively, these appear as brush borders because when you look at the microscope image, it looks like a brush stroke has appeared. In the last part of the small intestine, called the ileum, we find Peyer's patches in the wall of the small intestine, and those are nodules of lymphatic tissue. Also present in the villi are lacteals, which are important for the absorbing the products of fat digestion. So this image is showing the histology of the small intestine. And we can see here in this specific example that we have the layers of the small intestine shown on the top of the slide, the mucosa, submucosa, muscularis, with only 2 muscular layers, as well as the serosa. Looking at the bottom of this slide, we're going to notice that there are circular folds which occur, and Within these circular folds, on top of them are intestinal villi. On the right side of this slide, we're going to see a little more detail with the mucosa. So the epithelium are on the villi. So this would be one villus that we are seeing here. Within the villus, we have simple columnar epithelial cells with microvilli. And those microvilli have the appearance of looking like a brush stroke, or what's called a brush border. There are specialized cells that are found on the microvilli, things like the microvilli, and those are on top, or basically they're extensions of the plasma membrane of the simple columnar epithelial cells. And this is important to absorb nutrients. The goblet cells help to produce mucin to ease the passageway of the broken down foodstuffs, as well as I want you to notice the lacteal, which is in the center of the intestinal villus. This green, light green structure would be the lacteal for absorption of fat. The other cells are not important to remember. This is showing the histology of the small intestine. So notice for an intestinal villus here, we see an individual villus, and lining the villus would be the simple columnar epithelium with the microvilli, and you can see interspersed are the goblet cells. Shown on the right here is a much stronger magnification of the microvilli. These would be those microvilli, which look like brush strokes. This is an entire or portion of the simple columnar cell with the microvilli. And the microvilli are really just extensions of the plasma membrane. So it's part of the cell that's going to increase the surface area for the nutrients. So let's say, for example, that we have amino acids located here. Those amino acids can then be absorbed through the microvilli. On the image, the last image that you have on your slide here, we see an image showing the layers of the small intestine. This would be the mucosa. This would be the submucosa, the part that looks like it has kind of a light blue color. And then beneath that would be the muscularis layer. So you should be able to identify something like this on the quiz or on the exam.